Hey everybody, in this video we are going to talk about a really interesting story about how parental diet can influence offspring phenotype. Now, really cool study which integrates DNA methylation and epigenetics and gene regulation in eukaryotes. So a bunch of different things that we've been covering in this course recently, and also some things that go way back to the beginning of the course. We'll talk a little bit about meiosis and gamete formation and the agouti gene in mice. So this agouti gene is coming back, and we're going to learn a little bit more about it. So now we're going to start with mouse coat color. And mouse coat color, if you remember, is called agouti. And what is agouti? Well, if you look at a mouse hair, and some of this is review, the very tip will be black. You know, we're looking with a microscope, for instance. Tip will be black, and then we'll see a yellow band. And then black again, and then another yellow band. And I never realized this, right? Because we don't usually look at, I don't really see many mice ever, actually. But when I do, I'm not looking at their coats with their hairs with a microscope. I'm like, you know, running and screaming, hoping the cat or dogs will get it. Just kidding, I don't have mice in my house, so thank goodness. 1, 2, 3, 4. Okay, so we have here, and we don't get like a zebra pattern when we're looking at the mouse, because from far away, this gives sort of a brownish color. So these are black, these are yellow, these bands are yellow and black. So far away, it looks kind of brownish with maybe some black speckles. That's what I like to. Speckles are black, and that's what it kind of looks like to me from far away. And you know, if you want, you can take a look on the internet, take a look at just a regular mouse, a picture of a regular mouse, and you'll see, oh yeah, it's not quite all brown. It's sort of brownish with some speckles in there, some speckled pattern. This is the agouti pattern. And if you're interested, these pigments here, they have specific names, and these pigments are conserved in mammals. So even in humans, it's these mixtures of these pigments that lead to our coloration, our hair color and skin color and all that. I'm not an expert on that, but as I was doing some research on this, I noticed these pigments were coming up with reference to human hair color. So let's see, black is eumelanin, and the yellow is pheomelanin. But we'll just refer to them as yellow and black. Now, the mouse coat color gene, well, the agouti gene, is 131 amino acids, and in humans it's 132 amino acids. But we're going to just talk about the gene in mice, and we can assume it works similarly in humans, or has a similar role in pigmentation. So actually, you know what, I shouldn't say that. I don't know what the function of the agouti gene is in humans, but I'm assuming it has something to do with pigmentation, because it's a very similar sequence and length. So okay, back to the mouse coat color gene. Now this is what I know about. So we can have two wild type alleles. Let's just call these A. And if a mouse has two A alleles for the agouti gene, these are the wild type alleles, I could put pluses if I wanted to. The color would be agouti. And we could have a heterozygous genotype with A, little a being the null allele. These mice would be agouti also. And then we have two little a's here. The two little a's, these guys would be black. Those mice would have a black coat. Why would they have a black coat? Because this yellow pattern can no longer be deposited in intervals like this. So we just get the black pigment in the coat hairs. So you might also remember when we were discussing the agouti gene in the past that there's a well-known allele called AY. Now, this is a dominant gain-of-function mutation such that when it was heterozygous with a wild-type allele, the phenotype is yellow. So essentially, the yellow pigment is being produced all along the hair, or being deposited all along the length of the hair. There's no black. And we also learned that in the homozygous condition here, where both alleles are AY, now that's lethal. And there was a really good question at the time, and I didn't know the answer then, but I was able to look it up. And as I promised, I am going to tell you now why this is lethal. So scientists determined that this AY allele, how was it made? Well, let's take a look at the AY locus. So if this right here is the agouti gene, and let's say this is the agouti promoter right here. I'll put P with a superscript A to mean the promoter for the agouti gene. Over here is another gene. We're going to call this Rally, R-A-L-Y. We're going to call this the promoter for the Rally gene. Now the Rally gene is always expressed. So this promoter, it's always working, it's always expressing this Rally gene. Now, the AY allele, how that was created was all of this was somehow deleted. So from the end of the Rally promoter to the beginning of the agouti gene was deleted, and this Rally promoter was fused to the agouti gene. So all of this was lost. And the Rally promoter was fused to the agouti gene. Now this is the AY allele. So not only does it include a loss of the agouti promoter, but it also includes loss of the Rally gene. So, and the reason why this is a gain-of-function mutation is because the agouti gene went from being on sometimes to being on all the time, because this Rally promoter, it constitutively expresses, it always expresses the gene it's attached to. So that's why this is dominant to the wild type allele. And the mice are always yellow. But in mice that have this genotype, notice that a mouse with this genotype will not have a copy of the Rally gene, right? Because the AY allele, if a mouse has this allele, that means this allele is associated with the deletion of the Rally gene. And also, if it has the AY allele for the other copy of the agouti gene, it's also going to be lacking that Rally gene next to it. It turns out Rally is critical. This is a critical gene. So the mouse cannot survive without a copy of the Rally gene. So that's why mice with this genotype do not survive, because they lack a functional copy of the Rally gene. So the question way back from the beginning of the course was, how can a gene that just controls coat color or pigmentation of mouse hairs, be lethal in the homozygous condition? Well, it's not so much that it's the agouti gene itself, overexpression of that is causing the lethality. It's loss of the gene next door to the agouti gene. So that's a little bit of an aside, but it's actually important, because we need to talk about a different agouti allele that also causes a yellow coat color, but it doesn't cause lethality in the homozygous condition. So now that AY allele, we're going to call this one lethal yellow. So that's a well-known allele in mice. Mouse geneticists know this allele. This is a famous allele. Now they also have isolated other alleles. This one is called AVY for viable yellow. So this is an allele that is dominant to wild type and leads to the yellow color, but in the homozygous condition, it is viable. So AVY, AVY. So viable. Now the yellow color, this doesn't always lead to yellow. There's something interesting going on here, and that's what we're going to talk about. So let's talk a little bit about this structure right here first. So how did the AVY allele arise? So we haven't talked about these yet, but we're going to a little bit later in the course. We still have, I think, what, about a third of the course left? So let's say this is the agouti promoter here. Promoter, agouti. Here's the agouti gene right here. Now what happened was a transposable element. We haven't covered these yet, but we're going to cover them soon. Transposable element. And for now, think of these as selfish genetic elements that are found in the genomes of prokaryotes and even eukaryotes. I mean, different types of transposable elements in prokaryotes and eukaryotes. The ones in eukaryotes can jump around the genome and they can cause mutations. And in this case, a transposable element, we can abbreviate this TE, jumped in next to the agouti gene, next to it, so right upstream of the promoter. And inside that transposable element is a promoter-like sequence, so it can function as a promoter. Now, when this promoter functions, it leads to overexpression of the agouti gene. So the agouti promoter works, but when this also is working, it causes increased expression of the agouti gene, leading to that yellow phenotype. However, transposable elements are bad, and geneticists can debate this. So they're good for evolution, right? Because they cause mutations, and mutations can sometimes be good for, discovering advantageous combinations of genes, but for the most part, they're bad. And cells try to ID transposable elements and inactivate them. Because if they don't inactivate them, they can jump around the genome and cause mutations in critical genes. And cells, they don't like that. That's usually not a good thing for them. Inactivate them. And one of the ways they inactivate them is by methylation, DNA methylation of CpG dinucleotides. So I promise we're going to be bringing in things that we've covered very recently and things we've covered in the past. So DNA methylation of CpGs, DNA methylation of the transposable elements can inactivate them. And it turns out DNA methylation of this promoter region can influence that promoter and keep it from causing the overexpression of the agouti gene. So we can have, if mice with this AVY allele, this allele may be differentially expressed depending on the level of methylation of the transposable element here. Okay, if you're a little confused, hold on. I think I'll be able to bring you back with the next page of notes here. So, let me see. Okay, let's review this. Now, the transposable element, the promoter region, or promoter-like region, has CpGs. So if these are highly methylated, the agouti gene is expressed normally. if they're, let's say, not methylated. A agouti gene is overexpressed. So agouti gene expressed normally, the mice have the agouti phenotype. If this is overexpressed, the mice have a yellow phenotype. So what if there was a medium level of methylation? And then somewhere in between, so we'll say in between phenotype. Okay, so we have mouse coat color being a pretty good indicator of DNA methylation levels, at least with respect to that, with that transposable element there. So we have a neat experimental system there. And so what scientists were interested in, scientists who had been working with this system, they were interested in what would happen if you took female mice that have this AVY genotype and fed them a diet high in things like folic acid, vitamin B12, etc. And I'm going to say etc. because what they're looking at is a bunch of different nutrients that increase SAM levels. Okay, SAM, what is that? Well, SAM stands for S-adenosylmethionine. And now remember when we had talked about DNA methylation of cytosine, we just said that the DNA methyltransferase transfers a methyl group to the cytosine, but I didn't say where it took that, where it got that methyl group. Well, it gets it from SAM, the compound called SAM, so S-adenosylmethionine. And there are these nutrients that are thought to increase the levels of SAM in the cell. So what these scientists were thinking was, okay, so maybe if we feed female mice diets high in these things right here, we'll get more methylation of the AVY gene during gamete formation. Okay, so now you might be like, what are we talking about? Okay, so if you remember in the previous video, we talked about DNA methylation of the IGF2 allele during the formation of sperm in male mice. So it turns out that other genes can be methylated. At the beginning of meiosis, DNA methyl marks are early, early during gametogenesis. DNA methylation marks are erased from the genome. In male mice, some genes are remethylated. In female mice, other genes are remethylated. So the end result is, it turns out in female mice, this transposon next to the AVY gene is one that is methylated during egg production, oogenesis in mice. The AVY gene, or the transposon next to the AVY gene, is one of the methylation targets for methylation during oogenesis in female mice. So if this allele right here, this piece of DNA, represents the AVY allele in an egg cell, now it can be methylated. And what the scientists were predicting was that, okay, if we feed these female mice nutrients, lots of nutrients, like an insanely high levels of nutrients that are thought to increase SAM levels, well, then maybe these mice would produce eggs with high methylation levels of the AVY gene. And then maybe this would influence the phenotype of the offspring. So I have this, let me diagram this a little neater for you. So here's the experiment. we have female mice with the AVY allele, homozygous for the AVY allele. So we'll say female, and let's say these are going to get a normal diet. So normal for the nutrients thought to increase SAM levels. And we're going to put the dad over here. The dad is going to have 2 little alleles. So the dad's not even going to contribute a normal agouti allele. He's going to give a null allele to the offspring. So we'll say male, and the males are given a normal diet. And over here we'll have a different female. Now, female has the same genotype as the other female, but it gets a diet that is high in folic acid, etc. Very high, let's say very high in folic acid, etc. So what we're expecting, or predicting, I should say, with this experiment, what the researchers were predicting is that in egg cells, or eggs produced by these mice over here, that the AVY allele, right, because the mom can only donate AVY alleles to the eggs, the AVY allele will be, say, not methylated. We can go to the extreme here, not methylated. Over here, these females over here, which are fed diets very high in folic acid, well, the folic acid will lead to high levels of SAM. Lots of SAM will lead to lots of methylation of the AVY allele. So these are the egg cells, and the dad here just gets a normal diet and is donating in a little A allele, and it doesn't matter. He doesn't have the transposon here, so it's not going to be methylated anyways, even if it was methylated during spermatogenesis. So here's the sperm. Now, the prediction is that the zygote is formed over here. And what do we have? We have the AVY here, which is not methylated, and the A allele here. So the AVY allele, when it's not methylated, that transposon promoter works and causes this agouti allele to be overexpressed. So offspring are all yellow. All the offspring are yellow. Over here, okay, what's going on? We have the AVY allele. And it's highly methylated. Those are methyl groups. And then we got the A allele from dad. Now, the AVY allele is highly methylated. So as a result, the transposon that's next to agouti is having no effect on expression of the agouti gene. So the agouti gene is being expressed normally. So all the offspring are agouti. Okay, so that was the experiment, and this is what they set up. And did they observe this? For the most part, what they saw, and the results are significant, is that the mice from this cross over here tended to be yellow, and the mice from this cross over here tended to be agouti. So what they found was that, yes, indeed, the diet of the parents can influence the phenotype of offspring, and that influence is, in this case, is determined epigenetically. So very interesting study. I think this was back in the first decade of this century. But very interesting. And if I lost some of you there, I have typed this up nice and neat in the notes, so I encourage you to take a look at it. And if you have any questions, please email me. And I guess I will see you, I guess we have an exam coming up, but after that we have lecture 19, which we're going to be talking about, I believe, dosage compensation and I think sex ratios and some more interesting things. Okay, see you in the next video.